Thank you all so much for joining. Congratulations on making it to the 7th and the last week of the course. This is a wonderful accomplishment. You know, we've covered so many different topics ranging from introduction to carbonate chemistry to SDG 14.3, monitoring and managing data, cleaning it up. And then the last couple weeks, we've been focusing on carrying out experiments and doing monitoring. So we're very excited to welcome Dr. Helen Findlay and Dr. Steve Widdicombe with us this week to discuss Module 7, which is biological observations in the fields. But also, if you have any other questions about the course, they're both experts in the oceanification community and can answer a number of potential questions that you may have. So feel free to raise your hand or add those questions in the chat, and we can pause and address those as they come up. With that, we'll get us kicked off. Do both of you want to share a little bit about your work, where you are, and what you do?
Yeah, sure. I can kick off. So I'm Dr. Helen Findlay. You'll have heard from me throughout this module if you've watched it already. And so I'm based at Plymouth Marine Laboratory here in the UK. I am a biological oceanographer by training, so I did my undergraduate degree in biology and then moved into oceanography after that in my master's and PhD. So initially I started off doing oceanification research, looking at the impacts on biological species, looking at early life stages of barnacles and some of the other rocky shore species. And then kind of realized that actually we need to understand a lot more about how the organisms are related to the chemistry that they're experiencing in the marine environment. And so I kind of started to bridge the gap a little bit between the chemistry and the biology. And so I'm also now in charge of the carbonate chemistry monitoring here at PML and in the laboratory as well. So we've set up a whole raft of different experimental conditions in the laboratory, but we also do a lot of field monitoring and trying to kind of match up that biology and chemistry in the field as well. So.
Thank you so much. Dr. Widdicombe.
Hello, I'm Steve Widdicombe. I'm also at Plymouth Marine Laboratory. I'm a biologist, primarily interested in, well, my initial interests were very much in seafloor organisms, in benthic ecology, processes that sustain biodiversity and also the impacts of climate change on biodiversity. For many years, I was the head of the Marine Ecology and Biodiversity Group, but subsequently I'm now Director of Science for PML's science portfolio. So I have quite a broad remit ranging from Earth observations, modeling, ecology, biogeochemistry, and social sciences as well. My ocean acidification research was very much focused around experimental studies, looking at the impacts of elevated CO2 on marine organisms and biogeochemical processes as well. And I'm now also the co-chair of the Global Ocean Acidification Observing Network. So I have a strong interest in supporting and growing observations, not only of carbonate chemistry parameters, but also associated biological parameters that go along with demonstrating what impact is likely to occur alongside the chemical changes that we see. Thank you.
Fantastic. Thank you both so much for joining us. And we're really excited to hear more about your experiences and your expertise throughout the hour that we have as it comes out through answers to some of the questions that we have. But also thank you for joining us at this time as well. You may have mentioned, or you both mentioned that you're in the UK, so it's quite early/late where you are. So thank you again for joining us. We have a couple of questions just to get us started. And again, for our participants here today, if you have any others, feel free to raise your hand and unmute yourself or continue to add them in the chat. But one of the questions that we're curious about is, what's represented in some of the lectures. There's been a lot of work on biological responses for organisms that are in temperate latitudes or high latitudes, such as polar environments. For our participants that are coming from the tropical Pacific, do we expect different responses from organisms that are in these tropical or perhaps subtropical environments? And are there changes to how we then monitor the biological responses in these sorts of environments too?
So I can take a first stab at that, if you like. So I would actually argue that quite a lot of the work's been done in the tropics and subtropics as well, especially the coral work and some of the fisheries and organisms that live around the coral reefs. So if you're interested in coral reef work, then that's mostly been done in the tropical and subtropical, because that's primarily where the warm water corals are. So the short answer is, I think what we were trying to demonstrate in this course is that it doesn't really matter what species you have in your local environment, but it's the processes and the ideas behind the monitoring protocols that you can apply anywhere across the globe. And hopefully when people kind of are able to use those ideas to really look at some of these process changes, so calcification or primary production or changes in heterotrophic metabolism or growth, then those can be applied wherever you are across the globe. And actually that's really interesting then to be able to compare across different regions and across different latitudes and gradients. And that allows us then to kind of say, well, is there a kind of difference between the species that maybe live in the tropical areas? Are they more susceptible perhaps than some of the temperate regions? So there's kind of that element of it that we expect there to be, we do expect there to be differences, but the methodology and the techniques that we've kind of tried to talk about in this module should be transferable across all of that. But there's a lot of evidence to suggest that local populations and local acclimation really matters. So understanding the natural conditions and the variability of your local species or your local environment is really important to be able to interpret the biological response to what's going on and really understand what it is that's driving those changes in the biological response. So I think a key point really is to try and make sure that you align any biological monitoring with good chemical monitoring as well. I'm sure Steve can maybe add to that as well.
Yeah, I mean, I think you're spot on there, Helen. I think that second point you made was really critical. A lot of the impacts we see are fairly generic in terms of the processes that are operating sort of within the organism in which they respond to ocean acidification. But the really key thing is about understanding the environment and the context within which your subject organisms have been living prior to any experiments you undertake on them. It's so important to understand how they may have undertaken some pre-adaptation to perhaps elevated CO2 previously. The work published by Christian Vargas and others that explored different individuals or different populations from the same species but from different conditions showed very strongly how that local adaptation can not necessarily alter the direction of the response, but certainly the speed and the magnitude of any impact that you see. So I would strongly advocate that experimental studies should be supported by an appreciation of the conditions from where you take your subjects from, and also the conditions that you're then going to be applying that knowledge to when you take that out from the experiments.
I think that's such a good crossover between Module 5, that was Sam's lectures on laboratory experiments in the field, and this module right now, where we're talking about observations in the environment that are relevant to the organisms that we're studying. That's incredibly important to work on those aspects together. And as Alexis has just shared in the chat, the Go On in a Box kit, the opportunity that's available through this project, and the application again is open now. That's one way to sort of apply this kit, is gathering that sort of environmental data. So if you do have questions and want to bring organisms back in the lab, you have some sort of understanding of what your species or your population of interest has been experiencing. So thank you. With that, I'll turn it over to my colleague Meredith from NOAA to ask the next question.
Sure. Looking down on our list, it kind of flows nicely from talking about establishing a chemical monitoring program first as an important foundation, but many of our students are probably really interested in finding out what the biological responses are of the valuable species in their area. So, and we don't need a laundry list, but what would you say is the bare minimum to build up a biological monitoring program in terms of basic tools and instruments, and what sorts of spaces they may need to access? What sort of training do they need to take to get to that level? And of course, it doesn't need to be a laundry list, but what are some maybe unexpected things that you might need to acquire?
Do you want me to start with a general, Helen, before you go in? Okay. I mean, the key thing about biological monitoring, I think, is to think about what element of the biological system is relevant to the questions you want to ask in your particular context. So, example, if your driver is about understanding the impact of elevated CO2 on aspects of fisheries, or whether it's about biogenic habitats like coral reefs or seagrass beds or mangroves, or it's on primary production, or it's on harmful algal species. The key thing is about knowing what it is you're trying to achieve by doing that. It's far more powerful rather than just to think, oh, I'm just going to pluck a potential indicator species out of the air, to think about what is most relevant to the system that you're working in, and also what is most likely to be relevant to the skills to which you have access to as your own skills, all the skills and facilities and capabilities around your organization or colleagues you may have. What we've tried to do in a recent paper that we've submitted for publication is try to identify ways in which we can bring together all sorts of different biological observations clustered around a set of processes that we know are likely to be sensitive to elevated CO2. So rather than trying to set out and say, oh, I have to monitor pteropods, or I have to monitor coral reef abundance, the thing is to think about what capabilities do you have, what is sensible, what is reliable, because it's better to collect robust, reliable biological data that you know you can trust than try to endeavor to collect biological data that is of lower robustness or trustworthiness. So I would say, think about the key processes that you're wanting to get an idea about, be that calcification rate, be that production, be that levels of biodiversity, and pick a part of the system that you feel comfortable and confident that you're actually going to be able to collect high quality data to be able to address that question, and not feel that there's a gold standard thing to measure, and if you can't do it, then all your data is worthless, because there's all sorts of biological data that you can measure that is which is of great value to understanding how the biological response couples up with the chemical changes that you observe. Helen?
Yeah, I completely agree, and I think the modules, this module was kind of founded on that paper that you talked about, Steve. So once that's published, I think that will be a nice resource for a lot of the kind of go along with this module, really, in the fact that we were talking about calcification and primary production and heterotrophic indicators and genetic indicators, rather than looking at necessarily species specific responses. So yeah, just to kind of reiterate what Steve says there, is the importance of understanding your system and what it is you're looking for, and also just remembering that this is potentially needs to be done for a long period of time. I mean, you don't need to do it for a long period of time just to get an idea of your system and how it kind of maybe matches up with the variability in the carbonate system. But if you want to experience change and monitor that change over long periods of time, and thinking about maybe if your stakeholders are interested in putting in management techniques or management plans then you need to be able to maintain this monitoring for long periods of time. So it's something that your institute or your community can keep going as well. So you need to consider that as well, that the longevity of this ability to monitor as well. So yeah, I think that's probably.
Yeah, and I think if we think about how that kind of looks in reality, if let's sort of work through an example. If you're in a situation where you were worried or concerned about the health and function of a coral reef, and you knew that you were measuring ocean acidification parameters and wanted to explore some biological effects, you could think about, well, I'm going to measure the calcification rates, the actual rates that are occurring through time. But that's quite a complex measurement to make, and relies on certain pieces of equipment that you may not have access to. So that's not to say now you give up and say, well, I can't get a handle on calcification. Think about what else you could measure. And is it a case that it's about, could you pick some particular individuals of corals and map growth rates in terms of how quickly those corals are depositing calcium carbonate through time? Could you look at the percentage cover of calcareous species as opposed to non-calcareous species on the reef? Could you actually take samples of coral and look at the signs of damage on those species? So all of these different parameters are starting to give you an insight into the calcification and the bioerosion rates but they're not always needing to go right to the most complex observation of taking direct measurements of calcification. There are other ways to get a handle on how ocean acidification might affect calcification. And if we think about measuring biodiversity, obviously it would be fantastic if you could identify every individual in samples and enumerate all of those individuals down to species level. It's incredibly difficult to do that, particularly in areas where species descriptions are somewhat lacking and the taxonomic information isn't out there. But can you look at things like particularly well-described groups? So pick a subset of the community that you have confidence in being able to measure diversity. Or do you move up to a slightly higher taxonomic level and look at changes in the family compositions or the genus compositions? So I think the thing is to think about the process that you want to get information for, and then work out, what is a practical way in which you can gather information that tells you something about that biological process.
I think that's wonderful advice, and it goes back to what we were discussing in Module 5 about running biological experiments with Dr. DuPont, right? There's many different ways to run those, more complex ways to set those up, sometimes really expensive and time-consuming ways to measure samples. Might think about genetics, for example, is one way to do that, which was a little bit more discussed in this module too, but there's still value in going to some of those measurements of growth, of survival, of looking at damage, for example, that does contribute to the body of knowledge. So I think that's really appreciate that you think about how to build up to some of these more complex measurements, for example. Thank you. One thing that I wanted to go back to, Helen, you mentioned, when we're doing these sorts of measurements of processes, the practices are somewhat standardized globally, right? It's not latitude specific, for example. And I think that also speaks to the last lecture in this module about what to do with the data once you have it. And throughout this course, we've learned about the SDG 14.3.1 data portal to upload carbonate chemistry monitoring data and the metadata that's included with those sorts of measurements. And so I think there are a number of other options that you talked about in ways to deposit biological data to contribute to this more global understanding of these sorts of processes and oceanification. So I'm curious, from your perspective, in your experience, what sort of information is important to collect or document when you're out measuring these biological processes or collecting samples that are important to include when you're submitting it to one of the databases that you mentioned to ensure it's reproducible, or to add on additional context that's useful for when looking at these results in a larger context?
So I should I should probably start out by clarifying that I have I primarily use other people's data rather than collect a lot of this myself. So, for example, at Plymouth, we're lucky enough to have the Western Channel Observatory, which is our long-term time series station. And there you can look on the website and look at all the data available and the protocols involved in collecting those. So that's just another resource if you search for Western Channel Observatory. But so I think the important aspects really are the same sort of things for the carbonate chemistry. So you still want to be thinking about the metadata. You still need the quality assurance of the data. Like Steve says, you need to be making sure that the data is of a good enough standard quality. So there will be standard protocols for many of these types of measurements in terms of what kind of quality you should be achieving. But also, like the carbonate chemistry, and I think Adrian Sutton talked about quality assurance and quality control, you will develop your own knowledge of your own system and your own data and the machines and the sensors and things that you use. And so you can kind of develop your own quality control very much in the same way with the biological data as you would do with the chemical data. And it's important, I think, to characterize and have those uncertainty and quality flags incorporated with that biological data as much as it is with the chemistry, excuse me. And any auxiliary data that you have, I think, from my perspective, I think it's important to keep that data together. So if you have collected some physical or chemical data or other biological data, I don't see the harm in keeping that all together in one database, because actually that's incredibly valuable, useful information that you can immediately use and keep together, and for everyone else that can then make use of that data and share it. With the idea being that in an equal and equitable world, we will all be able to access each other's data sets. And so the more information you can put into that at the beginning and think about having, the better it is in the long run for inputting that. I was going to say something else and it's gone. My 1 o'clock in the morning brain has just lost that train of thought.
Not to worry. I think that advice is even more valuable when you mention working with others' data too. As someone who did a meta-analysis before of laboratory experiments, I realized just how important it is to detail certain notes in the manuscript, or what's important to go along with the data. So having your perspective of working with others' data and knowing what's critical is very vital too. Thank you.
If I just comment on that as well, I mean, metadata is so important to support the interpretation of what you're actually seeing. As we've gone through the journey of learning how ocean acidification is affecting organisms, it's been clear that there's been attributes and traits that perhaps weren't the primary interests that have been incredibly important. There were a number of experiments done a little while ago which actually started to explore the effect of the sex of the organism in terms of its response, and we could actually see a difference in response between males and females. And you think, why might that happen? Well, if we consider that the energy demands on the different sexes at different times in their life stages, particularly, the energetic investment in eggs is far greater than it is in sperm. So if you've got organisms which show sexual dimorphism, in the fact that you may well have organisms that have different sensitivities at different times of the years based on the timings of when you do the experiments. So all those little notes about, well, actually, if it's possible to differentiate between, if you're working in maybe decapod crustaceans, where you can say, well, actually, this portion of my test organisms were males and these were females. And I noticed that it was around the time of the year where they may be approaching, the mate breeding season, or they may be approaching the time in which they would normally molt, molting period. And also those kind of contextual data, when did you do your study? Where did you do your study? Starts to give us a real insight into some of those other aspects and contexts that might be influencing the results you get. Because it's no surprise to me that somebody could do an experiment on a species, and then somebody else could try and repeat that experiment using exactly the same species, but the result will be different. And in fact, it would surprise me greatly if the result was the same. The most important thing is to work out why those results were different. And that will tell you a huge amount about how the process is actually affecting and occurring within the individuals. Based on, maybe you got them from different populations, you did them at a different time of the year, they were a slightly different age or life stage, there's a different distribution between males and females. All these kind of things will mean that the results you get may not be identical to the results other people get. And I think, there's a lot of pressure now in the press about, I have to get the same result. If I do the same experiment as somebody else, I have to get the same result. Well, I don't believe that happens in ecology, because there are too many contextual variables that go on around those for you to get exactly the same response. The key point is to understand why those responses might be slightly different to when an experiment was done previously. And metadata and contextual data are so important for that.
It would be akin to assuming that if you did a medical study on every single person, you would expect to see the same response, and we don't. And it's quite clear that people understand that variability within medical science, but they don't quite understand that we get the same response in biological systems as well, and we do. And there's no reason why we shouldn't.
I think that's super helpful context. In the ocean acidification community, there's a lot of discussion around that with some of the fish studies over the last couple of years, and perhaps that's what you're speaking to. So I guess following up from that, I'm curious to hear from you also, for those of us who maybe do like the first experiment on a new group of organisms, perhaps a new genus or something that we're focusing on. How far do you think we can extrapolate those results? How, or where should we sort of bound our research to? Should it be describing like this group of animals during this season, during this time of the year, and that's what our results apply to? Or do you think there's a bit more flexibility on, thinking about what it might mean for a larger group that we're interested in?
I think the key thing there is when you're thinking about your experiment, be less interested in what happened and more interested in why you think it happened, because the what may not be you may not be able to extrapolate the what to other situations. But what you may be able to extrapolate is the why something happened. I mean, to go back to one of the very early experiments we did, and Helen will remember this well, when we were looking at calcification and arm regrowth in brittle stars in a calcifying organism. And our assumption was that these organisms we were going to take off some of their arms under high CO2 conditions and measure the rate at which they grew back, with the assumption being high CO2, the rate of growth coming back is going to be much slower in the high CO2 conditions. But it wasn't. These organisms responded in a way that was completely unusual to us. So rather than just reporting the fact this is what happened, our real question was, why did that happen? Why didn't it do what we thought it was going to do? And when we looked into that a bit more closely, we found that there were other effects within the organism, which was pointing to the fact that there was this transfer of metabolic energy from one process to another. So by understanding why we saw the response we did, it was much easier for us to extrapolate to other systems and say, well, if this process is happening here, why can't this process also happen in other species, in other situations, and in other contexts? And I think that's really clear really important. When you do an experiment, you see what happens, but you're really trying to get to grips with why that happened. And you can raise hypotheses about why you think, and sometimes they will be wrong. And that's what science is. It's about testing those hypotheses. And if they're wrong, then we need a new hypothesis to test. But don't be frightened to think about the why rather than just what happened.
If I just add, that kind of applies to the monitoring in the field as well. So trying to understand what's going on in with the biological changes that we might see in the field. It's not necessarily the what's happening, it's the why is that happening. So again, it's being able to match up to that auxiliary data, understanding the temperature regimes of the species, understanding the changes in the carbonate chemistry, any other conditions. I think when I started off in the introductory talk, I said, think about the context of your system. So is it near rivers? Is it going to be influenced by lots of land processes? Are there any mitigating or ameliorating factors that might affect that? And so the same kind of context applies. It's like, why am I seeing this response, as opposed to what the actual response is.
Excellent, thank you. And I think sometimes those situations are what makes being a biologist so exciting, is seeing unexpected results and getting to think about what's really driving that. Thank you, Meredith, for noting in the chat if anyone has any questions, if this is sparking any sort of interest, or you'd like to, ask a particular question about measuring some sort of process or something like that, feel free to add that in the chat. We'll get to it. But I'll turn it over to Meredith for the next question.
I'm going off script a little bit because your answers inspired me. You were talking a little bit about unexpected traits that, or places where you found unexpected results in a particular species. But I also wonder, as our students are thinking about their ecosystems around them, what are some surprising species or taxa that they might want to target? Like we always think about calcifiers, but that's really not the only taxa, the only type of creature that we should be investigating. So I'm sorry for throwing you a curveball, but are there any other surprises that our students should look for?
I would say there's value in any part of the biological system, because what we've seen is surprising results in a number of places we've looked. As an example, I spent a bit of time looking at the way in which ocean acidification affected the nutrient cycling in sediments. Because my first love was in bioturbation, in organisms that burrowed and created microhabitats within the sediment. And I was really interested to understand how these organisms that live under the sediment in pretty much high CO2 conditions themselves, how they were likely to be affected. Because the thinking was, well, what will a few 0.1s of a pH unit in the overlying water affect those organisms? Why should they be affected? And also, why would we expect to be affected in terms of the nutrient transformations that are driven by microbes? So I think microbes were traditionally thought of as being pretty tough and resilient, so particularly those living in the sediments. And what we actually found was that for some of these active bioturbators that create burrow systems in the sediment and actively irrigate those burrows, they were actually incredibly sensitive to the changes in elevated CO2 in a way that maybe not necessarily cause mortality, but would actually change behaviors in terms of things like burrow irrigation rates. And then the knock-on effect then is that the changing oxygen dynamics, because of the change in irrigation rates in the burrows, was affecting the way in which the microorganisms structured themselves between the anaerobic and the aerobic bacteria, which had a big influence on nitrogen transformations, the balance between nitrification and denitrification. So a process that we thought, well, there's no, how could ocean acidification possibly affect things like denitrification, which is an anoxic process living deep down in the sediment? We're suddenly seeing that changes in the ecological connectivity between different parts of the ecosystem can have huge effects on the way in which that ecosystem functions. So I would say, that there's no organism or biological processes or biogeochemical process which is particularly out of scope, as long as you understand how direct and indirect, or even indirect consequences can cascade through the ecosystem to cause effects. And that's the importance as well of trying to think in a conceptual model way, and thinking about why things happen. You know, we often think modeling is something that's done in computers and is a numerical process, and everyone has to program to be a modeler. But I would argue that everybody who's running an experiment is a modeler, because what you do is you create a conceptual understanding of how you think something works, and then you challenge that. And the direct effects of ocean acidification on some parts of the ecosystem might not be the things that are interesting. It might be the indirect effects. Another example is if you're affecting the behavior of one organism, the cascading effects of trophic interactions, of competition, of interference could well create effects on organisms that are potentially resilient to ocean acidification, but they are certainly not resilient to some of those ecological effects because of changes in the distribution or activities of their predators or their main competitors.
So I can just add another example of that is this kind of phytoplankton secondary production, sort of zooplankton. So we might expect that phytoplankton potentially increase in or don't have a negative impact necessarily directly from ocean acidification, but actually some of the work showing that the composition of the phytoplankton community changes, leading to potentially more abundance of smaller species or different types of species, which can then have knock-on implications for the secondary production, and how much of that energy is being transferred down the food chain. And also some of the literature has shown that the fatty acid and lipid composition has changed within those phytoplankton species, which again has consequences for the amount of energy that's available for the zooplankton communities. Even to the point where this is Sam Dupont's work, where he's been testing, he did a couple of tests on shellfish taste. So did consumer tests on whether prawns, and I think he maybe did it also on oysters, I can't remember, but he definitely did it with prawns, did some ocean acidification experiments on prawns, and then got some people to blind taste test them, and to the point where consumers could actually taste a difference between acidified prawns and non-acidified prawns. So there's all kind of surprising results that kind of have those knock-on implications because of these changes in the energy budgets and the lipid and fatty acid compositions of those sort of organisms. So.
And I would say this is why we don't want everybody running experiments on the same organisms, because the idea being that at the start of ocean acidification, you always had to run experiments on calcifiers because that's where the impacts were going to be. And it comes back to the point we made earlier about if you've got a particular species that's it's ecologically or environmentally important to your ecosystem, then have a look at that species. Don't think, well, it's not a calcifier, therefore I can't work on it, or it's it's potentially not going to be affected by ocean acidification. The more we learn, the greater breadth of subjects we use, the more we start to learn, and the more we learn from these surprising results.
And that's also true. Sorry, Meredith, you go. I was just going to say, it's also true for sort of monitoring again in the field. If we bring it back to that aspect, if we're thinking about some of the things that we maybe expect to see positive impacts from acidification, things like the seagrasses or the mangrove systems or things like that, again, it comes back to these sort of nutrient dynamics that Steve has talked about. So there may be consequences that we are not expecting that result from those communities being robust to ocean acidification, or actually almost ameliorating acidification in their immediate location. So we have to think about those ecosystems again, which is why it's important to understand what it is you're interested in monitoring in the field and really getting to grips with what's there in terms of the habitats that you're interested in and what you expect them to be doing in terms of acidification. So the seagrass beds, for example, have been shown to potentially ameliorate acidification locally, and any shelled organisms or kind of creatures that live within those seagrass areas or even on the seagrass blades have the potential to benefit from being within those systems. And so that's an important aspect to consider when you're thinking about monitoring within the field as well.
Yeah, I was just going to add that I love that study that Dr. DuPont did, because I think it's also very useful to sometimes choose species that are culturally important, that may understanding their vulnerability to ocean acidification would be very important for the community. And frankly, we should study them even if we don't think they'll be vulnerable, because finding that they would be resilient is also an extremely important result. And so, yeah, that's a wonderful study. And I wanted to pass it over to, well, Steve, you want to add something before?
Well, yeah, no, I think it's exactly right. But I think the key thing is when we find the resilient species or populations or individuals, you know, why are they resilient? And that's the really powerful thing. You know, finding resilient individuals within a pop. When we look at an experimental result, we often think about, if you say, oh, we had a 80% mortality rate, you know, and we report the 80% mortality as if that's the most interesting part of the result, but actually 20% of that population survived. Why did they survive? Why did they stick it out? What's so special about them? And actually, I think when you look at the variability around your mean result, it tells you so much about the variability that's in the population, you know, and tells you about the range of sensitivities from, you know, from the highly sensitive all the way to the vulnerable. So a null result, so to speak, you know, animals didn't die in high CO2 conditions, is not a boring result. It's an incredibly interesting result if you start to think about why that was the case.
Thank you. That's a great point. Always asking why, always. Caitlin, would you like to toss it to the next question? I was going to read a little more about the article that was shared in the chat, and we can also try to address that question as well, or the response.
Thank you. That's great. I do want to change gears a little bit and hear a little bit more about both of your experiences carrying out some of this work. You know, when I was first a graduate student, I had no idea what it looked like to go out and run an oceanification experiment or spend time in the field. It was just sort of a black box to me. So I'm curious if each of you could describe, you know, what this work might look like in practice. Like, for example, Helen, your colleagues that are going out and collecting some of the phytoplankton data around PML to contribute to these long-term data sets. What does the day look like when they trek out into the field and collect that data and bring it back to the lab? I don't think I hear your audio. Maybe it's just me though.
How's that? Is that
yes
back? So yeah, so like I said, at Plymouth we have what we call the Western Channel Observatory, which is a series of stations that we go out and regularly monitor. We have one which is station L4, which is offshore by about 7 kilometers offshore, I think. And we try and get to that station weekly. So that involves every Monday usually, getting the boat from the nearby harbour. We take, this is detailed, we take the van down from the lab, which has got all the containers in, ready, that are put out ready by the various scientists that are interested in collecting data that day. They all get put on the boat goes out to the site, and then there's a series of samples taken. So we do CTDs, taking temperature, salinity, and with depth. Then we have Niskin bottles, which we have, I think we've got a 12 rosette Niskin bottle case, I think now. So we can collect water samples for any of the processes that need the water. So that includes the nutrients that we collect. So we collect inorganic nutrients, nitrate, phosphate, silicate, things like that. The inorganic chemistry, so the carbon, dissolved in organic carbon and alkalinity. And other things that we collect the water for are things like HPLC or chlorophyll sampling, and We also collect water samples for flow cytometry. So that's looking for the nano and pico level plankton. And then we do net sampling. So that involves taking 2 different size nets. We have a phytoplankton net and a zooplankton net. And those are then lowered sort of from the bottom. We do a bottom to surface trawl of those nets, different net sizes to collect the phytoplankton. So all of those samples are then either preserved on the ship using whatever is the standard protocol for preserving them, or they're brought back live material to the lab. Usually that's done within half a day. So the samples tend to get back to the lab by about 1 o'clock at the latest for us normally, depends on the tides a bit. And if we can get into the marina. And then the scientists will go about either preserving the samples ready for analysis, or they will take them straight to the lab and run them through machines. So for example, the phytoplankton data, that's Claire Widdicombe's data, she will do microscopy analysis on Lugol's preserved samples. So they will go to run through and take microscope slides and go through the identification of those after, I think she filters them first. The same with the zooplankton, we do microscopy on the zooplankton. And then we also have a flow cam, which is like you were saying about taking pictures of these phytoplankton, zooplankton as they go past on the pier. We have a flow cam, which is a laboratory automated camera system to detect zooplankton species, which then the computer will sort of take a selection of pictures for, and we can match that then with the microscopy work as well. So yeah, it tends to be a long day. It takes some people get it all done on the Monday if the weather's good. Some people, the chemistry data, for example, we can be a bit more free when we actually do that. Don't have to immediately do it because we can preserve it. Yeah, so that data will then gets collated with internally within whoever's working on that data set, and then they go through the sort of the quality control procedures of it before submitting it. Eventually, usually there's some kind of time lag between actually getting the data and actually being able to submit it to a data center. I think we pretty much run on about 6 months to a year time lag between the data collection and data submission. If it's not already freely available on the websites, then it's available through the British Oceanographic Data Centre. So that's kind of a fast run through of what a day on the boat might look like. We try and do that weekly. Obviously, that's weather dependent. And then we have other sites that we do bi-monthly, so twice a month if the weather's good. But like all time series, that means that we do have gaps in the time series resulting either from weather or from loss of ship time. Either the engine broke over winter for us for a good number of weeks, so we had to try and either get other people to help take samples or we just missed out on the data. So these are practicalities that happen everywhere, and you can see those kind of time periods through the data where maybe something's happened. Either you've had big storms, you can't get out to sea, and sometimes they're the most interesting times that you want to be able to measure, but you can't necessarily get there. So that's something that you have to just think about as well when you're analyzing and interpreting the time series station, that it's not necessarily always the most complete. But then moving forward in time, I guess, in looking into sort of future technologies, this is kind of as a side, there are going to be developments, and there are developments on automated sensors, things that allow us to maybe fill in some of those gaps. So things like you say, these sort of in situ camera technologies that can take photos of phytoplankton, zooplankton as it moves past the mooring is fantastic. And to be able to match that up with maybe pH sensors or PCO2 sensors on a buoy would be a really nice thing to have going on. So yeah, I don't know if Steve wants to add anything.
No, just a few things really. I mean, the new technologies are certainly improving, and things like eDNA as well is giving us a new insight into biodiversity through a different methodology. It's never underestimate the amount of work involved in generating high quality time series biological data. You know, it can take a matter of minutes to take a water sample and then days and days to work through to turn that into species abundance and identity data. So it is a huge amount of work, but incredibly valuable. I was somebody I know who runs time series said to me, you should never start a time series and you should never end one. But I wouldn't want people to listen to the first piece of advice, because I actually think that they are exceptionally powerful and valuable things and much underrated. Biological and chemical time series data are like gold. They are so valuable, but they are incredibly time consuming to generate, and it takes a long time to really get the true value out of them. They can take, you know, the longer you run a time series, the more valuable they become. So it's difficult in a situation whereby a funder might expect quick results and quick wins from an activity. But if you've got a funder that's in it for the long game, then that's fantastic. But they are difficult to find.
That's fantastic to hear about, just the process of going out and what that looks like. And then, maintaining that for a long period of time. It's truly an effort, but it's been at least so beneficial for my work and many of others too, who have benefited from these long time series, to understand the conditions and build off of that when we're, doing a small project that may perhaps, work itself off of those sort of data. So it's really great to hear about. We do have just a couple more questions or minutes left, but I have another question for Steve. In addition to doing all this excellent science that you've shared with us today, you also have a leadership role in the Global Oceanification Observing Network, which I know you contribute a lot of time to. We learned about GOA-ON a little bit during week 2 when we discussed PI TOA, the Pacific Islands Hub for GOA-ON, but I want to hear from you, what have you found that's been really valuable to go just outside of your research and work in this sort of community? And why would you recommend our participants here today get involved in this global effort?
Yeah, certainly. I mean, joining the GOA-ON network has been immensely beneficial. I mean, from a scientific point of view, in terms of, we spoke an awful lot over the past hour about understanding the context and actually the power of bringing multiple data together and multiple viewpoints and perspectives and insight together. And something like GOA-ON as a network is a really fantastic tool to be able to bring the community together and bring different perspectives that you start to see the whole picture. Often with our own research, you only see a little small part of it, and you can almost go back to the old Indian story about the blind men who were trying to describe what an elephant was by just feeling little bits of it, and they all came up with different answers depending on which bit of the elephant they were actually able to get hold of and get an appreciation for. But by talking together, they had put it all together and realized what it was they were dealing with. So I would say that the real power of a community is about sharing perspectives, also sharing lessons learned, providing support in terms of abilities and capabilities. I think the beauty of GOA-ON is some of the things that have been spun up in terms of the peer-to-peer process, and very much in peer-to-peer, it's about matching up people with particular skills that they might be able to help somebody who has a particular skill need. I would never like to see peer-to-peer as a way of saying, oh, this is an established researcher helping an early career researcher. It's more about matching up skills with needs than it is about trying to put early career with late career researchers together. And having a network to be able to do that, I think, is really important. The other great thing about GOA-ON is it's given the community a voice that it far outweighs any voice we might have been able to have as individuals. the influence that GOAN has had on international stages, such as the UNFCCC COP meetings, far outweighs what we would ever have been able to achieve individually as researchers. So the power of joining a network is not only about what you can get from it in terms of knowledge and information, but it also increases the amount of influence you can have and make changes and make decisions at national and regional and international levels. We saw this year how the ocean was finally recognized in the final decision documents that came out of Glasgow, out of COP26. And that's only because networks like GOAN and other strong networks are able to create that influence and represent a voice for the science community itself.
That's incredible. Thank you. You've made such a contribution to the community. So thank you both for joining us here. It's fantastic to have your expertise not only in the lectures, but being able to have this discussion too. And just reflecting what Sally added in the chat, it's very appreciated you joining us. Throughout this course, we've talked about how great the oceanification community is. And I think that's been no truer than seeing some of our lecturers join, in the middle of the night, because they're really excited about being able to share and continue to contribute to those that are a little bit more newer to the field. So again, thank you. As we wrap up, I'd also like to congratulate all of you participants for making it to the end of our 7-week course. Just as a reminder, all of the quizzes are due by April 25th, so make sure to get those completed. You'll also see 2 surveys open up next week. The first is a complement to the pre-course survey that you took. It's the post-course survey, so similar questions. And then you'll also have an opportunity to reflect on the course. This is the first time we've run this Ocean Teacher Global Academy course, so you can provide your feedback on how we can improve it for future iterations that the Ocean Teacher Global Academy will be able to host. So again, thank you all for joining, and have a great rest of your day.
Thank you.
Thank you, everyone. Bye.
Thanks, guys.